Hello everyone. Welcome back to Dr. Liu's VCE Biological channel. Today, let's learn together about the 2020 VCE Biology. We will focus on questions about DNA manipulation techniques and applications. My explanations may need to be revised. If any teachers, parents, or students find errors, please feel free to provide your guidance. This question is about PCR. Pairing primers with complementary nucleotides is a chemical process that does not need any enzyme. RNA polymerase catalyzes the synthesis of RNA, not DNA. Annealing of primers occurs at 40 to 60, and extension of the DNA occurs at 72. So, option C is correct. The number of copies of the DNA is doubled in each cycle. A bacterial plasmid was modified by inserting a gene for an enzyme that provides resistance to the antibiotic ampicillin. This plasmid is transformed into E. coil. Then, these treated and untreated E. coils are cultured overnight. The untreated E. coil is used as a control. The next day, the bacterial cells are spread on 4 agar plates. Plate 1 – untreated bacterial cells on nutrient agar. Plate 2 – untreated bacterial cells on nutrient agar with ampicillin. Plate 3 – treated bacterial cells on nutrient agar with ampicillin. Plate 4 – treated bacterial cells on nutrient agar. The plates were incubated overnight. Drawing a diagram helps thinking. Since plate 3 contains ampicillin and the plasmids resist ampicillin, only cells taking up plasmid can survive. The process in which the bacterial cell takes up the plasmid is called transformation. After the target DNA is inserted into a plasmid, there is a gap between the target DNA and the plasmid. To link this gap, DNA ligase is used to rejoin the sugar phosphate between the plasmid and the target DNA. Rice is a staple food for billions of people worldwide, particularly in Asia. Although rice supplies energy, it is low in micronutrients, such as iron and zinc. Australian scientists created a strain of biofortified rice that has been trialed in the Philippines and has been recently introduced to Bangladesh. The table below compares the iron and zinc content of normal white rice to that of biofortified rice in ppm. The biofortified rice was created when 2 particular genes were inserted into normal rice. The biofortified rice plants responded as if they were iron deficient by permanently switching on another gene to take up iron and zinc from the soil. This technology will increase the concentration of zinc and iron. So, it is to improve the nutrition of malnourished people. 2 exogenous genes are induced into the plants. So, the biofortified rice is genetically modified and transgenic. Gene cloning is applied to manufacture large quantities of proteins at a low cost. In the past, before the development of DNA technology, proteins for treating certain health conditions could be obtained only from animals, such as cattle and pigs, or from human corpses. State 2 advantages of using gene cloning to manufacture pharmaceutical proteins rather than sourcing the proteins from animals or human corpses. These advantages are constant and reliable supply, purer or more effective products, larger quantities, less chance of side effects such as allergies, and more appeal to patients rather than using animals. Outline 1 ethical issue associated with the use of gene cloning in the manufacture of a pharmaceutical product. The suitable answers are who has the legal ownership and how will the product be made available for all people in the world, changing a species' DNA may result in unforeseen consequences. Concerned that the pharmaceutical product may contain bacteria that will cause disease, and not natural, and therefore may be against religion views. A student wanted to investigate the effect of two different endonucleases, restriction enzymes, on a linear DNA fragment. The student used 3 tubes containing a buffered solution of linear DNA fragments, each fragment being 9,500 base pairs in length. 2 different endonucleases were available. BAMH and HIND are added to tube 1 and tube 2, respectively. Both BAMH and HIND are added to tube 3. After incubation, the DNA from these 3 tubes is measured with electrophoresis. The results of electrophoresis are shown here. Please analyze these results. 5 marks cover 5 points. Shorter fragments move further and longer fragments move less. Standard used as a comparison and reference. BAMH cuts the DNA fragment in one place and produces 2 fragments. HIND cuts the DNA fragment in 2 places and produces 3 fragments. The largest fragments are produced when cut by HIND. BAMH fragment lengths are 4000 bp and 5500 bp, and HIND fragment lengths are 8000 bp, 1000 bp, and 500 bp. When the 2 endonucleases are present, 4 fragments are produced. Sizes of 5500 bp, 2500 bp, 1000 bp, and 500 bp are produced. The student repeated the experiment the next day. This result is different from the previous day. The absence of bands for BAMH may have been caused by incorrect loading of the DNA sample into the well. Only 1 band for HIND may have been caused by the restriction enzyme being denatured. Some factors influence the rate of movement of DNA fragments through the agarose gel. These factors include the size or length of the fragments, concentration or viscosity of the agarose, and voltage or power used. However, the length of time does not influence the rate of movement of DNA fragments. Outline 2 safety guidelines that should have been followed by the student. These safety guidelines are making sure the electrophoresis equipment is turned off to avoid electrocution, wearing safety glasses to protect the eye, wearing gloves to avoid contamination by DNA or the chemicals being used, and correct disposal of materials. In DNA manipulation techniques and applications, 6 multiple-choice questions are worth 6 marks, and 2 short-answer questions are worth 15 marks. The overall score for this section is 21 marks. With a total possible score of 120 points, the percentage for this section is 17.5% of the total score. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.